Welcome viewers. Today we are going to see the pulse field gel electrophoresis. It is one of the important techniques nowadays used in research and also in clinical labs. First, before going into this, we should know something about the basic about the pulse field gel electrophoresis. What is this electrophoresis? This electrophoresis is a very powerful genotyping technique used for separating a large DNA fragments. Normally, if you go for any electrophoresis, they will separate the DNA fragments less in size. Whereas this technique is used to separate the large DNA molecules greater than 20 base pairs. So normally these large DNA fragments will be getting it after digesting it by cutting with the restriction endonuclease enzymes. Then you take this large DNA fragments, apply to the gel matrix under the influence of electrical field. It is moves. But what is the important thing in pulse field gel electrophoresis is that this electrical current which we apply for separating the DNA fragments, we are applying in a periodical way by changing the direction. So it is a variation of Agrove's gel electrophoresis that permits analysis of bacterial DNA fragments over an order of magnitude larger than that with what we do with conventional restriction enzyme analysis. So it provides a very good representation of the entire bacterial chromosome in a single gel, which can be highly reproducible profile, so that we can clear distinct and well-resolved DNA fragments we can get. First, we'll see about the principle about the pulse field electrophoresis. The principle of this electrophoresis revolves around the use of alternating electrical fields to separate large DNA fragments. That is a very important thing. Here we apply the electric current in a different way compared to other electrophoresis where it is very uniform electrical field. But here we are using alternating electrical field to separate the DNA fragments. So the key features in this electrophoresis is two things. One is alternating electrical fields. Other one is pulse electrical fields. So normally we know that we have a constant electric field is applied to separate DNA fragments, but here we use alternating electrical field. This electrical field periodically changes in direction, thereby the molecules, the large DNA molecules, reorient themselves and migrate to the cell matrix. So this mainly applying this alternating field strength mainly prevents the DNA from migrating too slowly, allowing for the separation of very large DNA fragments. The pulse electrical fields, we know that we are applying alternating electrical field, and this alternating electric field is applied in the form of pulse. So during each pulse, what happens? The DNA fragments moves to the gel in one direction. After that, we go for another pulse. There is a pass. So this pulsing mechanism helps to ensure the DNA fragments move as far as possible without running out of the gel due to the large size. This is very important about the principle about pulse gel. The instrumentation part, we have electrophoresis apparatus. There is a large horizontal gel electrophoresis chamber so that they can accommodate a large agarose gels. So to this agarose gel, we'll be loading the DNA samples that will be strained with the DNA binding dyes for visualization. We have two electrode sets. Here that is specialty in this electrophoresis. So these electrodes mainly generate electrical field. But what is the main difference between these two is that we have two sets which supplies current in a pulsating manner or in different cycles, alternating cycles they will do. Instead of one set of electrodes, we have two sets of electrodes in this. Then we have a controller for the pulse. That is a very important component of this instrumentation because this regulates the duration, intensity, and the timing of the electrical field pulse. So we can program this controller so that we can create desired pulse pattern to separate the DNA fragments effectively. Then we have a cooling system because due to this current, they generates heat and the DNA should be moved to the gel properly. So in order to prevent the gel overheating and maintain a consistent separation conditions, we use a cooling system. So they mainly present in the form of circulating buffer cooling bath that is used to remove the heat from this electrophoresis unit. Then after we run this electrophoresis complete, then we separate the bands visualized by staining it by using ethidium bromide or cybergreen. So then the gel is typically photographed and imaged using UV or blue light to document the DNA patterns. So regarding the setup, what we do, how we work, carry out this electrophoresis. First, we prepare the DNA samples. That is genomic or plasmid DNA of interest. What we do, we digest it with the help of enzymes. Then after digesting, we get a large DNA fragments. Then we cast the gel, agarose mostly used. Then to this agarose, we use these samples to be loaded. Then the gels are normally in this pulse field gel electrophoresis, we use agarose gel of larger size compared to our agarose electrophoresis. Then after loading the DNA into that wells in the agarose gel, then the gel is placed horizontally in the chamber. Then the two sets of electrodes, which is positioned at either side of the gel chamber. Then we generate electrical field necessary for the separation. And afterwards we apply alternating field because for effective separation of the fragment we apply alternating electrical fields. Then we have a controller to control the pulse or timing, duration, intensity of electrical current we are giving. This is very, very important. Then because of this current heat is generated that should be dissipated by cooling system we are using it so that we maintain a very stable temperature to prevent overheating. Then afterwards running that, then the DNA fragments is taken out, then they are strained to see properly, visualize properly, then we can analyze. So this is how we do it. That is, we cut the DNA using precision base. Then afterwards we see these two sets of electrodes that is responsible for moving the DNA in a zigzag fashion by the separation of heat. Likewise, we can see here how it moves. In a different manner, we are giving the pulse so that the DNA is moved in a zigzag manner so that the separation is very clear. So what is the application of this? It's mainly used in molecular biology and genetic research for a variety of applications, mainly genome mapping, that is to determine the size and the arrangement of DNA fragments within a genome. Genome typing, mainly to differentiate between the strains or individuals based on the unique DNA banding pattern, mainly used in epidemiological studies. And also to create DNA fingerprints in forensic science, and also for typing for bacterial strain. To investigate large-scale DNA rearrangements such as separations, inversions, translocation, duplication, all these things. We can use this pulse field electrophoresis. So detecting related strains in case of any epidemiology or pandemic, hospital outbreak. That we can, for all these things, we can use this pulse electrophoresis. The limitation for this is, main thing is very expensive and time consuming and we need a very high skilled person. Sometimes we cannot distinguish between the strains, difficult to interpret. And moreover, it is suitable for potential outbreak in hospitals spanning relatively very short period, that is 1 to 3 months. It is not more than, if isolates, analyzing the isolates, if it is greater than 1 year, we cannot analyze using this electrophoresis, this type of gel electrophoresis. That is one problem, disadvantage of this. The bands of same size do not guarantee the same DNA. So relatedness is only relative. That is a important limitations with this pulse gel electrophoresis. So with this, we have seen the outline about the pulse gel electrophoresis, the principle, the instrumentation, the working, the application and limitation. Thanks you once again for listening to this.